Welcome back. We are going to work on defining and explaining the importance in innate defenses as far as diapedesis, chemotaxis, opsonization, and a membrane attack complex. We'll get to each of those kind of along the way. We're going to describe the steps also involved in phagocytosis. We're going to look at phagocytes and how they do their process. So this is on your outline, page 1, Roman numeral 3, internal defenses, cells, and chemicals, and we're looking specifically at phagocytes. So we want to, these phagocytes are in our bodies. They kind of act as janitors and the police force. In other words, they're around to clean up any kind of debris that's available, and they're going to respond to invasions. There are a couple different types of phagocytes. They're called phagocytes because they do this process of phagocytosis, and we'll look at that in just a moment. We have big ones, they're macrophages, and we have smaller ones that are microphages. The macrophages typically originate from a monocyte. So remember when we looked at white blood cells, one of the types of white blood cells were monocytes. We're not going to look at how they convert from monocytes to macrophages. I'm just interested in you knowing that that's where they came from. Macrophages are either fixed or free. If they are fixed, that means that they actually, the cells themselves are actually in a tissue. They don't move around at all. An example of that would be the microglia in your central nervous system. The microglia don't float throughout your central nervous system. That probably wouldn't work so good with all the action potentials going on. But instead, they're kind of hooked in with the astrocytes and everything else as part of the neuroglia. And as something floats by, they'll have cytoplasmic extensions that will come out from them, kind of engulf whatever they need to and bring it in. The free macrophages roam. So a lot of times they're not necessarily a whole collection of macrophages in any one tissue, but these macrophages roam from one area to another. Macrophages found in the liver are called Kupffer cells. That's K-U-P-F-F-E-R, Kupffer cells. And so they circulate throughout the liver and get rid of debris. Again, they deal with toxins and anything that looks like something that doesn't belong there. They'll help get rid of that. And in your lungs, you have alveolar macrophages, which are sometimes called dust cells. So again, when we're breathing in, we have a lot of ways of filtering out that air. But if anything ends up down in my alveoli, in my lungs, I have these alveolar macrophages that are going to help me get rid of it so it doesn't build up in my alveoli, it doesn't cause an infection, we don't end up, it helps us resist the disease that might come in there. The macrophages are white blood cells that you looked at when you did your mini test, neutrophils and eosinophils. Both of these circulate throughout the blood. However, they will leave the bloodstream, kind of squish their way out through the capillary walls at the site of an injury or an infection. Neutrophils do this constantly. Eosinophils do it when the pathogen has already been recognized and has antibodies already stuck on it, and then that'll attract an eosinophil to come in and take care of things. As far as how these free macrophages move, and macrophages as well, either one of these, they move by a process called diapedesis, and let's look at these pictures. Move to the next one. So here we can see across fixed macrophage, we've got those neutrophils and eosinophils, which are macrophages. We have a free macrophage, which could be a Kupffer cell or one of those alveolar macrophages. And then you have the monocyte that these macrophages originally came from. Okay, so anyway, these guys, these free macrophages right here, are able to squeeze through these capillary walls, so they can actually push through. They're flexible enough in their shape that they can push through the walls, and that's called diapedesis. What causes them to get pushed through the wall is a process of chemotaxis. So these phagocytes are attracted to the chemicals that are secreted at the site of an injury or an infection, and that causes them, those secretions, particularly cytokines, it's a type of secretion, and that causes them to get pulled into the injury site or the infection site and start doing their job there. Opsonization, we talked a little bit about last week. This is going to help to attract the macrophage to a pathogen because it's already been coated. The pathogen has already been coated with antibodies and complement proteins, and we'll talk about those complement proteins in just a bit in the lecture series. Once a macrophage has come in contact with a pathogen, then it goes through this process of phagocytosis. The first thing that has to happen is adhesion. And adhesion is when the macrophage receptors attach to the cell membrane. And so that's kind of what you're seeing in here. It doesn't show it really well, but there would actually be receptors right here along the edge, and they're going to attach to this pathogen. And it causes the pathogen to come in as a vesicle. So the second part of it is a vesicle forms. This vesicle then fuses with a lysosome. So here you can see they're coming in contact with each other. Here they've fused. And then the enzymes that are in the lysosome is going to cause the digestion of the contents of the vesicle. Now, occasionally a macrophage will come up against something that's too big. It can't actually undergo phagocytosis. And if that's the case, it does one of two things. It'll bind to it and then wait to be assisted by other components of the lymphatic system. Or macrophages can also release a cytokine called tumor necrosis factor, or nitric oxide, or hydrogen peroxide, and all three of these can then damage whatever the macrophage is attached to. So it releases these various chemicals to damage whatever it can't swallow itself and hopefully destroy that pathogen in that form. The next type of innate immune system is undergoing immunosurveillance. These are the natural killer cells. We've talked about these already, but I just want to come back to these. These natural killer cells are a lymphocyte. However, they are not antigen specific. What they will do is they have a list of all of the antigens that belong to the body, and they will attack any antigen that doesn't belong to them. So they will attack cancer cells, and they will attack viral-filled cells. So we're going to look at the natural killer cells, and we're going to discuss their function. If we take a look at these natural killer cells, to me these are just amazing little cells. So the first thing that has to happen is recognition and adhesion. So again, we have these receptors on the membrane of the natural killer cells. We have the antigens on the abnormal cells. So these would be your class 1 MHCs. And this natural killer cell looks at its laundry list, realizes that nope, this antigen isn't on here, and it adheres to this either cancer cell or viral filled cell. Once it does that, its Golgi apparatus realigns so that it is directly across from the cell that it's attached to. The book refers to this as being like if you were standing by a tank and suddenly the tank aimed its gun barrel right at you. So this Golgi apparatus is swinging down and comes right in contact at the point of attachment. And then the Golgi apparatus releases a chemical called perforin, and you can see in this bottom part of the illustration that perforin then actually creates, this molecule creates a hole in the abnormal cell, and by doing that it causes lysis of the abnormal cell. Basically wreaks havoc with the osmotic balance of this cell and causes it to simply explode because it fills up with fluid and just simply explodes. All right. The inflammatory response. How does the inflammatory response... Oh, I'm sorry. I skipped a part on your outline. Let's go back to that one. Number 3 underneath natural killer cells asks you to think about natural killer cells versus tumor cells. So we have tumor cells in our body probably all of the time. We're really not aware of how often things don't quite go the way they're supposed to. And typically a tumor cell produces tumor-specific antigens. So the class 1 MHCs put out this tumor-specific antigen that our body recognizes and immediately destroys. Those natural killer cells see it and go through this whole process that we just talked about. However, some cancer cells are able to hide. In other words, they don't put out these tumor-specific antigens. And if they don't, then we have what's called immunological escape. There's no way for the natural killer cell to recognize this cell as not being part of self, and so it allows it to continue to grow, and it'll divide, and it'll develop tumors and cause all kinds of problems in our body. All right, I think looking at the time, we're going to stop now with this part of the lecture, and then we will continue on with the inflammatory response in the next lecture. Thanks for listening.